Hello everyone. This is a tutorial on how to use this ImageJ macro. This is the macro for the Mitochondrial Fission Finder. A tutorial video for performing image analysis. Let's first take a look at the file structure. This file structure consists of 3 channels. The first channel, when I open it up, alright, the first channel shows the mitochondria staining. The second channel displays the staining of the mitochondrial fission protein DRP1. The third channel is actually the protein we are interested in studying. So we want to know about this protein. When mitochondria undergo fission, its distribution in space and time, and its relation to the fission event. How are they correlated? Now, let's take a look. Besides being composed of 3 channels, it is actually an image with a long time axis. At the same time, you'll notice that, as the image is captured, its brightness gradually decreases. So, during our post-acquisition image analysis, some corrections need to be made. In this large set of image data, we need to extract the mitochondrial fission, that is, to identify the fission events. This is not something that can be easily discerned by the naked eye. And that is why we developed this macro to perform this analysis. Alright, let me explain first. After this macro is executed, what will we see? Firstly, as we mentioned earlier, our images tend to fade over time, becoming gradually weaker. So, in our real data, we will first extract the mitochondria from the first channel. For this part, after taking out the stained sample, you then proceed with bleach correction, so that all the images along the time axis have a consistent brightness. Next, after a series of image analyses, each mitochondria is assigned an ID, and then they are compiled into an object list, which is used to describe their contours and positions. So, in ImageJ, after performing this image analysis, you will obtain mitochondria IDs. And this is their list of ROIs. Within the ROI list, if you click on one, it will tell you the location of the corresponding mitochondrion. Its identification method is actually to matching the contour from the current time point to the contour at the next time point, so that it becomes obvious that at the previous time point, the so-called mother mitochondria was just a single, distinct structure. But at the next time point, you'll see it marked in 2 different colors, with these 2 different colors indicating that it has actually divided. And such results will be listed in the analysis results table, which will tell you the parental, or daughter, mitochondrian's ID is, and also, the time at which this occurs, and how many daughter mitochondria it ultimately divided into. Alright, let's see how to carry out this analysis. Let's close this file first. For a simple demonstration, to speed up the demonstration, first, I actually start by using the tool here, the selection tool, to select a small area. Then click on Duplicate to duplicate the entire dataset. Then it will crop out this area. After cropped, you can then save it to the hard drive. Remember to save it to the hard drive, because later we will automatically create a result folder in the same path and store the process data and the analysis results in that folder. So if your image has already been exported, make sure you save it to your hard drive. Alright, now let's analyze the file I've already exported. Okay, so the first thing is go to our GitHub space to download the Mitochondrial Fission Fibers ImageJ file. Download it, and then open it in the image folder. Next, I'll clear out some data from the hard drive. Okay, this is a set of data already stored in our hard drive. Just drag it in directly. Alright, after dragging it in, just press Run, and it will start running in the background. Once it finishes, it will tell you how much time this set of files took in total. Alright, the analysis is complete. So now let's take a look. What other results did it also produce in that folder we just mentioned? This is our file, in the same directory, where you'll see a result folder. Inside this folder, there are a total of 3 or 4 TIFF files. Wait a moment. I'm going to open it with ImageJ. We are going to open it with that. Okay. This is channel 1. This is channel 2. This is channel 3, because our file has a total of 3 channels. And here, the colors actually represent a series of consecutive events. This is the time series of the recorded parental mitochondrion. Therefore, set this as T here. Set this as T plus 1 here. That is, they have already fissioned. So, these are the mitochondria, and they have already fissioned. Sometimes we want to know, because the protein we are currently interested in is trino-3, this unknown protein. I also want to know, where is its location before division? So we also take the copy of image at T1. So, what you are seeing now is T1, the timeline of T and T plus 1. The contour lines drawn above. The 2 contour lines at time points T and T1 represent the positions of the parental mitochondria. The position at time point T plus 1 represents the location of the fission daughter mitochondria. If you want to observe how channel 1, channel 2, and channel 3 each behave spatially at the same location, you can go to the Analyze tools. There is a Synchronize window here. After clicking on Synchronize window, you can then synchronize all 3 windows simultaneously. Then you will realize that you can actually allow them to do so. They can be moved together or to make other adjustments. This helps you clearly identify the specific position or time point you are currently viewing. So, for this one, we can see that, because channel 2 is DRP1, we already know that DRP1 forms a structure that constricts and pinches off the mitochondria. As you can see here, at this location, where mitochondrial fission is about to take place, DRP1 really just happens to be right at this position. That would be at their junction. Meanwhile, at the T plus 1 time, you can observe 2 additional mitochondria distributed at this position, which seems likely to be the site of upcoming division. Okay, this is where you can check the file we have currently selected. This table basically sums up the events you're looking at right now. So how do you interpret the numbers on it? In the results folder, you can find the mitochondrial labels. When you hover your mouse over this label, you will see a number in this place. The numbers above actually represent the IDs of the mitochondria. So the labels for the daughter and parent mitochondria correspond to these numbers. That only represents an ID. If you also want to know their actual locations, the ROI list is saved together with results as well. So here, you can find out which mitochondrion this ROI list corresponds to. You can all see this yellow line here. Okay, this is the macro we're using. The introduction to the macro mitochondria fission finder.